This is the panel discussion with all the participants from the previous session. So, gentlemen in the panel, please pay attention. Sorry, I'm sounding more and more like a
She's a
teacher.
bad influence on me.
Okay. The first question is, again, I would like to keep it as a big picture question. Um, I know we're trying to promote discussion, we're trying to break silos and foster collaborations. What do you think should happen to make sure that it actually happens on the ground? And I still see a lot of people working in silos, and there's not really, we saw this yesterday as well. Four different people working on this with the same goal in sight, but different approaches without any communication. So how do we make this communication happen across different groups? How do we facilitate your research? Apart from that, I wish I had. No, money doesn't do it all, right? Money and?
Yeah.
Sure, but then money doesn't foster communication. The more money you have, because let me tell you a small incident that I faced in my life. So I used to work with the big pharma, and then it was an outsourcing model, and I was working with one of the the Ivy League pharma. I don't want to name names on the stage, but then they were trying to give me the same project, two different groups side by side, physically located in the facility. They were giving me the same project. And then when I looked at that and they went back to their inventory, their books, and realized that they were ordering the exact same chemicals, they were recruiting these people with the same background and similar expertise, and they were doing the exact same research without realizing that they were doing the same thing. Yes, within the same company. But what I'm saying is they had all the money they wanted. So money is not the only solution. Money is definitely one of the solutions, one of the big solutions. But money is not the sole solutions.
You better repeat what they're saying, otherwise people can't.
Yeah, here.
I think this ARPA H could be absolutely incredible if it can bring people together and provide them funds, and people, they're going to fund large groups, and I think that's it. That's what happened with DARPA. Basically, we had large groups and we all worked together, but you can't work together if you don't have.
But did DARPA solve ARPA-H or DARPA solve all the problems? I don't think so. They did make a dent in the.
Well, you have to have the right people that support the right questions. But if you're asking about four people that could work together.
Okay, so why I'm asking these questions is because we would definitely put it all in black and white, and we'll put it out there for people's consumption. So it's important to ask these questions and get the perspective from everybody here. So, Evan, your 2 cents.
So, one is having meetings like this, where people really, in a small, kind of an intimate setting, are able to hear each other and talk and socialize over dinners and drinks and things of that sort. So that's one way to do it. The other, I actually do agree with your comment about the funding, not the money aspect, but when RFAs are put out that emphasize multidisciplinary collaboration, that I found is an amazing catalyst, because at the end of the day, everybody has self-interest. And what I've noticed, and this has happened to me personally, NIH has helped a little bit when they went, there's such a thing as a multi-PI R01. So now all of a sudden you get people from different disciplines coming together, and it's not as if one has to surrender their autonomy or their ability to get a promotion. by being a PI on R01, now everybody can be equal R01s. Lately, our institute has been putting out many internal RFAs where you must have people from different disciplines in your group doing different things. And when that RFA gets issued, all of a sudden people in my institute who never talked to each other went, well, we got to go for the money. And it's not that much money. It's more the notion of, I mean, we're academics are pretty easy. We'll jump at anything that gives us any kind of money. It's not a lot of money, but that brought us together. We just put in just a letter of intent, a few-page letter of intent, investigators at our institute who would never have ever thought about collaborating. It was somebody in the gut, somebody in the lung, somebody in the brain, looking at looking at barrier function in different organ systems, you know, and we made it up because we wanted the money.
Question, did you get it?
It's still being assessed.
Interesting, I just saw a movie about the life of Michelangelo by a Russian director, whose name I can't remember. A remarkable film. The name of the film is Sin, S-I-N, just like a sin, right? But it's in Italian. It's an Italian-Russian co-production, all filmed in Italy, and you know exactly what it was like back then. So, back in the Renaissance, you had literally living in one small town, because Florence was a small city. Michelangelo, Leonardo da Vinci, and Raphael. Can you imagine living in a town of maybe 30 or 40,000 people? That was a population back then, okay? And having those three individuals cross each other's path. Okay, the three greatest figures of the Renaissance. But they did not work together. The point is they hated each other. Okay, they wouldn't talk to each other. If they spoke, it was always through a third party. Okay, but they each created their own school of art. I mean, Michelangelo is very different than Raphael, was very different than Leonardo. So, in a sense, what they probably did is they avoided a groupthink, okay? If they all got together, they would probably, their paintings and their sculptors would tend to look alike, okay? So while they were ultra competitive, because the church was supporting different artists for, paint the Sistine Chapel and, carve a statue of Moses, carve a statue of David, right? But they were in intense competition.
Yeah, but that's why having people from different disciplines working together is really good, because there's no competition.
Because there's no, in fact, competition.
You get help from a different point of view,
Right.
and it wouldn't be competitive.
So for instance, if you're looking at a system where you need the drug release is very critical, you need the kinetics. So you have a biomaterials person, but who is not a cell biologist. These two people are not ultimately competing because they're not in the same space, but they're synergistic because you're getting a controlled drug release, okay? And those collaborations tend to stay. But when you have two people who have the precise same skill sets, well, what happens is the question becomes, what's yours and what's mine? Where did that idea come from? Did I say it first or did you say it first, right? As opposed to cross-discipline, right, where you don't get that natural competition.
So I would really like to maybe point out something that might be impossible, might be sort of dreamlike, but ideally collaboration would be the way to move forward. Not so much what's mine, what's yours, what can I lay claim for, because then I can get money for it, because that's not helpful to promoting science. So even if I'm an expert in one thing and I talk with somebody else who's also an expert in stem cell, in my stem cell biology, we still don't think exactly the same. And these grant funds that are encouraging people from different institutes to come together has led to some of this. I haven't received funding from any of it, but it started collaborations where we say, okay, I have these resources, this is how I think, how can we best utilize these resources and how can we share them? And I think that's maybe a benefit that I have coming from the National Institutes of Health, but it's something that is very much trying to be promoted at the NIH to say, how can we use these resources to help other people and collaborate without.
We should hold on to.
We'll share one.
So maybe we should finish
So,
the panel.
yeah, I was in immunology, and so NIAID, NCI, NIA, all of these institutes had groups doing T-cell immunology. And everybody was doing the same experiment. Like they would hear, and they didn't talk to each other because they wanted to get to the answer first or whatever. So actually, it was really tough at NIH. I don't know what it's like now, but it
Maybe
was.
it's what's been fostered recently. I'm, just new to the NIH. Been there for 7 years.
And what happens is you get a convergence of ideas. Everybody starts working on the same thing instead of, starting to really think in different ways, independent of each other.
So I think it
Oh.
depends. It depends on both competition. For example, longevity as a field has so many of these competitions that go on, and they create huge developments and create milestones in the field. So I would love to hear more from Isabel.
Is this on? Yeah, I'd like to pick up on your analogy, and I'd like to see a collaboration or competition even between benefactors, because that's the common thread for your three high achievers, where the benefactors were competing to have the best artist.
There was a fight between who wanted Michelangelo to come to Rome, but the Medici insisted he stay in Florence. So there was these fights, like who was going to get Michelangelo, right? Who's going to get Leonardo? The big shot in Milan wanted Leonardo to come because he also knew how to make war engines, and he was very interested in, war engines. So, Leonardo said, well, if he's going to pay me for war engines, I'll go there. But there was competition between the funders. That's what it was. Competition between and when you have excellence, you track that competition.
So the benefactors are the companies who's going to commercialize your results of your scientific work. The benefactors, there's very few companies here. It's mostly nonprofits I see here that are supporting you. There are very few companies here who have, because the pharmaceutical companies are basically controlling everything, right? So those are the benefactors. If he's truly right, you need to have some corporate backing that's not just in it for the profit, but has the same goals that you have to be more philanthropic with your scientific outcomes. Does that make sense?
Yep.
Definitely. So, okay.
I don't, I guess I have a very skeptical view of corporate involvement. And I think nonprofits, you can induce them to collaborate and pool the resources. And I've seen unnecessary competition between the nonprofits for rare childhood diseases when there's more foundations than there are kids with the disease. But companies, and Jyothi maybe can comment on this, the more and more I've become involved with the entrepreneurial world, the more they want to keep things within house. They want the IP, they do not want to share, they don't want to collaborate. So I think that's my skeptical view. Maybe Jyothi has a different take on it.
I think if one positive change that's happened in the field of longevity in the last 1.5 years is there's a lot of money that's come in. Close to more than $2.5 billion has come in to this field. And 10 years ago, I always find this amazing that 10 years ago, we didn't have VCs in the venture capital funds in this field. Today, we have more than 30 venture capital funds, and then more and more people are interested in the field of longevity. For example, some sovereign funds are coming in as well, like Saudi Arabia and Kuwait, Qatar, Qatari Foundation is interested. So I think what is needed, I mean, this could be too much of an idealistic view, is to get the research that's happening out there, to get the right kind of visibility and to frame things in the right way. Because as scientists, I used to wear the science hat for a very long time in my past life, and then we have a tendency to frame things that only scientists understand. So if we can get out there and frame things differently that appeals to, say, maybe the patient advocates, maybe the venture capitalists out there, I think the money will be made available. So, and yes, sorry, just one more point. Yesterday we were having a discussion, Evan and I and another one of the other speakers that was here yesterday. She talked about this example of how publications are done in physics. So in biology, we have first author, the communicating author. Somehow there is a pecking order in each research, but apparently in physics, everybody is an equal contributor. So there is nothing like the first author, corresponding author. Yes, if somebody has received a grant, all the names would be on the authorship, but then everybody contributes equally. I think it has its own merits, and competition also has its own merits. So I would like to close this with 2 minutes more, and then possibly we can move on to the next question.
So I had the benefit of both systems, competition and I will say compassionate collaboration, if that's the word I can use. So my competition began when in 1995 or 96, as I was, talking to my friends, I wanted to do mitochondria, and then I realized after editing a book that it was such a field where it needed a lot of attention. So I thought, okay, let's just start so I started a journal, edited a book, and then I thought what next I can do. So I said, okay, let's bring a professional organization of scientists and physicians together to talk about this problem. And at the time, there were 1,100 journals which would publish mitochondrial articles. So I went to just a starting foundation on mitochondria, and I said, hey, I'm going to do this, and can you give me $500 to register this society? Later on, I learned, said, well, survival of fittest. We do not have $500 for you. But that competition, what led me to start the professional organization. Bob started mitochondrial medicine. We worked together, and that has become a copycat from around the world. So we have in Taiwan, China, Japan, Korea. So that was a competition which led to, otherwise if they given me $500, I would have done something, okay, no, everybody got motivation. But at the same time, I got this compassionate collaboration, which money was not involved, as you said, but money will work if we, and I find my personal experience is, unless there is a trust in the collaborator, you can do whatever money bring, it will still not work. So these type of conferences, small conferences, I met many new people here, meeting and helping and talking, build the trust so we can build on that. In the compassionate collaborator, I wrote an article about COVID and how it can hijack host mitochondria. I put it on LinkedIn. Somebody contacted me from King's College London. They said, hey, you got the idea, we got the patient. Do you want to work? We did. And we can clearly show that how this COVID-19 pathogenesis is due to hijacking of the mitochondria. And then another collaboration, and I have not met these people whom I worked and published. Another one was looking at mitochondrial DNA moving to the nucleus. The plant people were doing beautiful work on chloroplast movement in the nucleus. I sent them a letter and said, look, can you be kind enough to help me? And they say, yeah, why not? We will. And we get our technology worked out. So I call that compassionate collaboration because somebody thought value in it. They didn't ask for money or reagent or even at the end of the authorship, they said, well, you decide. So
Thank you. You have.
Continuation of the commercialization idea was, okay, the sales department isn't the whole company, but the whole company better be the sales department. So maybe the Healthspan that's putting all these nonprofits together should have a PR marketing salesperson using social media like you were found, and that's what's brought all these dollars into the longevity business. Look what it's doing to Budweiser, right? And Target. So we use the power of the new Healthspan organization to get the right marketing PR people behind the whole effort of the whole scientific community. Makes sense? Then make the companies really start salivating.
Wow. What have you done, Isabel? See?
Sorry.
What have you done? Anyway.
15 minutes. Let's go.
So he was, the joke is he was saying that he spoke to Isabel for an hour and we became 3 times smarter. So with all his comments, I'm asking Isabel, what has she done? What has she created? Evan, I want to move on to the next question. Cell therapies are very, very expensive, right? So you're coming up with another cell therapy using ESLs and...
I'm not ESLs.
No, you talked in the morning about NESLs, right?
No, they weren't ESLs. They were primary neural stem cells, very cheap.
Okay. So what kind of price points would you look for in this if it were to ever get to the clinic? I hope it gets to the clinic. So.
Well, you're probably talking to the wrong person because I actually am pretty left-wing and believe in socialized medicine, so I think the government, it shouldn't cost patients anything. But the therapy I talked about is actually inexpensive. Most of the costs are already involved in what we do anyway for the care of sick newborns in the NICU. Now that would be great to do something about making intensive care for newborns, intensive care for any age, more affordable. But then that starts getting into a whole argument or a whole discussion about who should be paying for health care in general, which is a little appropriate to the topic here. What we're talking about is how health care should be administered from actually pre-birth all the way into old age, and who should be paying for it. Even if we come up with a great drug, Isabel comes out with a great drug that deacetylates, that inhibits HDACs and things like that. Who should be covering all of these expenses? I think, and it was a point that was actually made a few days ago in one of the panels, is that at the end of the day, if the government would be more mindful of preventative medicine and then timely interventions with new drugs and new therapies, the cost to society would actually diminish the cost of hospital care. So I think that's an interesting way to go, and I'd like to think about that. There's a lesson actually, and maybe we can discuss this, that there's a way now to treat a kid's disease called spinal muscular atrophy, SMA, that formerly was, some people know it as Wernicke-Hoffman disease if you're thinking about your old training, that absolutely was lethal. You never made it out of infancy. And through AV9 now, there's a way of putting in a good copy of a gene called SMN that now cures the disease, or at least we think it cures the disease. The company that now is making this available now has the most expensive drug ever on the market. millions of dollars. And the argument is, well, if you had a chronic disease, you'd be paying for the lifetime. We're giving you something that cures the disease, but we still want a return on our investment. So we're going to charge a lot of this money.
Yeah, it's interesting because Provenge, one of the cell therapy drugs that came out for prostate cancer, was priced at $400,000 per dose. So that's what I wanted to discuss.
Well, I was involved, I and a friend. They were going to give an award to a particularly successful Wharton grad, okay? An annual Wharton Award, right? And I'm going to name names. The guy who founded a company called Spark Therapeutics, and their first product was a siRNA that inhibited macular degeneration, okay? All it required was a single drop in the eye, once or twice. I don't know if there was a second, right? And the way the president of the company said it was, well, we're going to set the price in the following way. How much is your vision worth to you? Okay? Right? How much is it worth to you not to be blind? Right? And not only did he do that, but he actually was charging for each eye. So, if you got a single drop in your left eye, that was $250,000. But that same little eyedropper that would have held another drop into your right eye was another $250,000. Okay? All right? And that was his marketing, that's what he learned from Wharton. Okay? Well, I and my friend, we said, listen, we're not going to, we squelched that. He did not get the Wharton Prize, okay? I said, this is the kind of person, right, that, you start thinking Stalinist thoughts, so to speak. So there's that aspect of modern drug development, which is almost, sort of like the airplanes now. In order to get if you want a boarding pass, that's another $25, right? Okay, put a bag overhead, another $45, right? It's so totally monetary that you almost vomit. Yet at the same time, you have to have the profit motive, right? So it's this balance, and I think sometimes embarrassment, right, is like a very powerful tool. How dare you, sir, charge for one drop $250,000 in the left eye, okay? But another drop, one more drop, okay, in the right eye is another $250,000. I mean, how about a volume discount, okay? How about maybe $10,000 just for the second drop, right?
Thank you.
Yeah, I have a brief comment on that. So we started out our NRF2 induction with a proprietary compound, and the company who owned the license for it through multiple communications said, no, we're not going to let you use that, even though it worked. So I have converted over to a non-proprietary version, which in combination I think will still be patentable, but at a $10,000 or 10,000-fold lower price. Right. Well, if they succeed.
Now, the other side, I can make this Talmudic discussion, like one hand on the other, is that the R&D, so let's go back to the SMA story again with AAV9. The research that was involved in getting there, is 3 decades of paying for PI salaries and tech salaries and postdocs and grad students, and at some point, somebody's got to pay that back. Now, I don't like the idea that some kid's family needs to pay $2 million because of 30 years of research, but at some level, which is why I start thinking that this is something that, this is what governments are meant to do. The primary goal of government is to ensure the health care of your populace. So I think that ultimately they should be covering all of this. But that's why I'll never be elected to the Senate. So.
There's another question from here.
Thank you. This one's for Dr. Singh. I enjoyed what you wrote or what you discussed about the mRNA DNA, or excuse me, the mitochondrial DNA repletion with wrinkles and solar lentigines. Have you looked at it in conjunction with energy-based devices, IPL, et cetera, or other forms of dyschromia that are hard to get with energy-based devices?
No, we have not, but I have certain ideas how we can do that. Obviously, you can look for some wavelength which can stimulate the mitochondrial function. And in the context of skin, I think that will depend on whether we want to target epidermis, dermis, or another part of the skin system. But certainly it's in our mind to do that. And I will be interested to talk to you or somebody who may be willing to help us.
We just have another 5 minutes on the panel. Yeah, so I do have a question for Isabel, and then I'll come over to you, yeah. So Isabel, you talked about various types of measurements in the hematopoietic compartment, right? So my question to you is, if you were to measure aging in various parts of the body, most of these aging clocks and other measurements all focus on blood. Is that a good enough representation, or we need other composite measures to look at biological age in the complete human system?
Thank you for that. So I do like to say, because I'm a hematopoiesis person, that yes, blood is the end-all be-all. It's easy to collect and will give us the most information. But even within the hematopoietic system, we know that there's various different aging processes. Our stem cells are not the same as our differentiated cells. We know that the epigenetics of them is different during aging. So I do think that as we develop these processes, the blood was a good starting point. But as we mentioned, all of these stem cells have different burdens. All of our tissues have different turnovers. So we'll need to be more specific about how each one of these processes age. There will be things that touch everything, like mitochondrial changes. There may be some consistencies, but I think each tissue type is going to have unique challenges and unique aging phenotypes that we'll really need to understand. And I don't believe there'll be one cure-all for everything, and that we'll need to uniquely tailor our either interventions or our treatments for the cell types that we're interested in.
Was your question for Isabel?
No. That was for Dr. Snyder.
Okay, we'll come back to Dr. Snyder in a minute. So, Greg, my next question is for you. Thank you, Isabel. My next question is for you. We earlier talked a little at lunch about intracellular energetics and how that could be the next potential therapeutic angle. Could you elaborate on that a little, please?
We have preliminary evidence from mass spec analysis of plasma and red blood cells, extracellular, intracellular differences. And one produces a marker that's better for showing brain amyloid load, and the other one produces a marker that's better for showing cognitive performance or cognitive capability. So these are different compartments, and I mean, if you just think of, what's controlling plasma, of course it's the whole body, but also a lot the liver. What's controlling intracellular? It's every differentiated cell in the body, and we know from parabiosis experiments that there is an effect from old plasma and young plasma. Very remarkable experiments.
Thank you.
I had a clinical question for Dr. Snyder. I'm an internist, so I was wondering, I so enjoyed your talk for babies and anoxic injury. I was wondering how that might translate to grown-ups with strokes, acute strokes.
I think it's applicable. Yeah.
I
I think stroke, different etiology, but very similar
We can take it off.
very similar pathophysiology.
Yeah, we can take the rest of the questions offline. I just have one last question for Keshav, because Dr. Kennedy I think he has time limitations and he's the next speaker. Keshav, you talked about primary ovarian insufficiency as the primary indication that you're going after. From what I'm given to understand, there are no oocytes left in that condition, and the only resort currently available is donor oocytes being used. So if you were to interfere with the mitochondria or enhance the number of mitochondria, is it even possible to relieve this condition, or is that the way to go about creating a therapeutics for this?
Certainly, the key is the early diagnosis. And what happened with those 30 million women I was talking about, they get diagnosed at a very late stage. And that is something, first, appears to be genetic. So unless they get a whole genome sequence, and which we can do now very quickly, one can at least predict that over time, there is a window of opportunity where you can perhaps protect, but that's yet to be seen. But certainly once the oocyte is gone, you can't do anything about it. But certainly, there are people I know, at least in mice, they've been very successful in doing so.
Thank you. So I'm going to end this because we need to move on to the next session. Sorry, I know there are a lot of questions with this panel, but then maybe we need to take it offline. Thank you.
Thank you.